Hello everyone, I'm Dr. Ashish Guragai, and today we'll be talking about innate versus adaptive immunity, and then we'll be talking about B lymphocytes and T lymphocytes. We'll be focusing on their development and their activation. So let's get this started. So let's start with innate versus adaptive immunity. So what is innate immunity? Innate immunity is nothing but our body's defense mechanism against all the pathogens that we encounter from minute to minute in our daily life. So why do you think we don't get upper respiratory tract infection all the time? Or why do you think we don't get skin infections all the time? It's because we have this innate immunity components such as physical barriers, for example, skin and mucosa. And we have ciliary motility and cough reflexes, which prevents us from getting infected by inhaled pathogens. So when we inhale a pathogen, ciliary motility and the sputum traps the bacteria, and then ciliary action expels the sputum, which has the trapped bacteria inside them. Similarly, the cough reflexes also help us to remove these pathogens from invading our respiratory tract. Now, the cellular components of innate immunity are macrophages, neutrophils, dendritic cells, natural killer cells, and complement proteins. Similarly, we have secretions like our saliva, which contains lysozymes. Similarly, we have gastric acid secretions in our stomach, which protects our body from being invaded by the pathogens that we consume. Now, the components of adaptive immunity are T cells, B cells, and the circulating immunoglobulins. So let's talk about the response to pathogens in innate immunity. This response is non-specific, which means our components of the innate immunity, they cannot distinguish whether this is bacteria, whether this is virus, or they cannot distinguish whether this is gram-positive bacteria or gram-negative bacteria. All they do is they recognize that these proteins or these antigens in these pathogens are foreign and they are trying to invade our body. That's all innate immunity does, all right? They know these proteins or antigens are foreign and we have to destroy them. That's all they do. All right. And the response they create is non-specific. Now, another important thing about innate immunity is that these reactions occur rapidly. All right. So let's say you get a cut in your hand. All right. Inflammation occurs within minutes. Similarly, because of the disruption of the physical barrier, that is skin, now the bacteria can invade our skin. But do we get an infection after every cut? No, right? We don't get an infection after every cut. Why does that happen? Because neutrophils and macrophages will rush to the site of inflammation and any bacteria which invaded our skin will be phagocytosed. All right, and this happens within minutes to hours. Now, talking about memory response in innate immunity, there's no memory response. That means our innate immunity does not remember whether Neisseria caused infection in our body or there was a streptococcal sore throat. All right. Our body does not remember the pathogens. All right. Now, let's talk about adaptive immunity. The components of adaptive immunity are T cells, B cells, and the circulating immunoglobulins. Now, compared to innate immunity, the response to pathogens in adaptive immunity is highly specific. For example, if there is a streptococcal sore throat, there will be a specific response to that particular strain of streptococcus. All right, it will be highly specific, and that response won't be same to another bacteria. It will be completely different and unique. The immunoglobulins that were produced against one strain of streptococcus won't be same compared to another strain of streptococcus. So that's why adaptive immunity is highly specific. But the thing is, this specific immunity develops over long periods of time. It does not happen rapidly. And during that duration, it's innate immunity which protects us from the pathogens. All right. During the development of adaptive immunity, we need some kind of protection, and that protection is provided by innate immunity. Now, unlike innate immunity, the memory response in adaptive immunity is vigorous and quicker, right? In innate immunity, there's no memory response, but in adaptive immunity, the memory response is really robust. Now, what does that mean? It means if we get infection from the same streptococcus after, let's say, 2 months, our response will be really quick and we don't need a long period of time to adapt. The response will be robust and there'll be specific immunoglobulins to destroy that particular strain of streptococcus. Now let's talk about pathogen recognition. So we talked about how macrophages and neutrophils go to the site of inflammation, but how do they recognize bacteria or pathogens? How do they recognize, okay, these are not our normal cellular components of our body. They are foreign. So how do they recognize it? Now, this happens from toll-like receptors. So toll-like receptors are a pattern recognition receptors. That means they recognize different proteins or different molecules in bacteria via these toll-like receptors. Now, let me give you a specific example. So toll-like receptors can recognize pathogen-associated molecular patterns or PAMPs. All right. Talking about gram-negative bacteria, they have lipopolysaccharide. And our toll-like receptor recognizes lipopolysaccharide and knows that this is a foreign pathogen, this is a bacteria, and the body knows that we need to destroy these pathogens. Similarly, there are other PAMPs like flagellin, nucleic acids, right? And our body is able to recognize these proteins via the toll-like receptors. Now, in adaptive immunity, this pathogen recognition occurs via immunoglobulins. Each immunoglobulin has a unique binding site to a particular antigen, and when that antigen is exposed, our immunoglobulins go and bind to that antigen and then trigger adaptive immunity. Similarly, like immunoglobulins, there are cytokines which are able to recognize the pathogen and initiate the adaptive immunity. Now let's talk about functions of natural killer cells. Now you might be wondering why I am only focusing on natural killer cells which are part of innate immunity, but there are other cells as well, right? But the reason I'm focusing on NK cells or the natural killer cells is because from the exam point of view, these are super high yield, all right? So what are natural killer cells? They're nothing but a type of lymphocytes which secretes porphyrins and granzymes to induce apoptosis of virally infected cells or tumor cells. Now, this image shows how natural killer cells identifies a pathogen and then releases porphyrins and granzymes. Now, if you look at this image, you can see that there are immunoglobulins which are binding to the antigens, right? Now, this will form an antigen-antibody complex, and our natural killer cells in the body are able to recognize this antigen-antibody complex, and then it will know that it needs to destroy the cell, right? This way, natural killer cells release porphyrins and granzymes and cause apoptosis. Besides this, natural killer cells are also able to recognize pathogens via PAMPs, right? Now, the activity of natural killer cells are enhanced by interleukin 2, interleukin 12, interferon alpha, and interferon beta. Now, these are super high yield information. Now, from the exam point of view, how they will ask a question about this activity enhanced by these interleukins is that they'll give you a question of viral infection. They'll explain how a kid got virally infected. And then they will say that natural killer cells were able to destroy this virus infected cell. Which of the following interleukins enhance the activity of natural killer cells? So this is how they ask question, and there'll be different options, and you have to choose one among these four. The high yield ones are interleukin 2, interleukin 12, interferon alpha, and interferon beta. So these enhance the activity of natural killer cells, all right? And these are super high yield from exam point of view. Now, there's one more really high yield information. These cells are also able to destroy any cell which lacks inhibitory signals such as MHC1 on the cell surface. Now, these are really important in tumor cells. Tumor cells, if they lack MHC1, it will signal natural killer cells that these cells do not have inhibitory signals. So these are definitely some kind of abnormal cells and natural killer cells need to destroy them. All right. Now, these inhibitory signals are really important because these signals protect our normal cells from being destroyed by natural killer cells. All right. Similarly, natural killer cells play a huge role in type 2 hypersensitivity by destroying cells via antibody dependent cell mediated cytotoxicity. We'll be talking about these in detail in our hypersensitivity video. As it is really important, I want to highlight this destruction of tumor cells by natural killer cell one more time. So the tumor cells, they lack MHC1, all right? And MHC1 is an inhibitory signal. If this inhibitory signal is absent, then it tells natural killer cell that this cell is some kind of abnormal cell and it needs to be destroyed, all right? Super high yield, that's why I repeated it multiple times. I want you to really remember this function of natural killer cells. Now let's talk about major functions of B and T cells. So these are just a basic overview of the function of B and T cells. So B cells, they initiate adaptive immune response via humoral mechanism. All right, the humoral immune response of the adaptive immune system is initiated by B cells. Similarly, they recognize and present antigen as well. The most important function of B cell is to produce antibody and also to maintain an immunologic memory. Similarly, the functions of T cells are to initiate cell-mediated immunity. There are different types of T cells. The most important ones are CD4 T cells, which helps the B cell to make antibodies and also produce cytokines. Similarly, there are CD8 positive T cells, which directly kill virus-infected and tumor cells. Now, T cells play a huge role via cell-mediated immunity to cause delayed hypersensitivity, which is also called cell-mediated hypersensitivity. It is a type 4 hypersensitivity reaction. Now, T cells also play a huge role in acute and chronic cellular organ rejection. So this was just a brief overview of function of B and T cells, and we'll be talking about them in great detail as the video goes on. But this was just an overview for you guys. Now, before we talk more detail about function of B and T cells, we need to understand HLA gene complex and MHC proteins. So our body has these gene complexes called HLA, human leukocyte antigen. So these gene complexes, they code for MHC proteins, major histocompatibility complex. All right. Now, MHC proteins, they play a vital role in initiating immune response as they present antigen fragments to T cells and bind to T cell receptors. Now let's look at this figure. This is MHC class 1. It has alpha 1, alpha 2, alpha 3, and a beta 2 microglobulin. All right. They are also called HLA, HLA-B, and HLA-C. All right. And this is the cleft where antigens bind. Now let's look at the structure of MHC class 2. They also have similar antigen binding cleft, but their components are different. Unlike MHC class 1, MHC class 2 has two beta components, beta 1 and beta 2, and they have two alpha components, alpha 1 and alpha 2. All right, they're also called HLA-DR, HLA-DQ, and HLA-DP. Now let's talk about MHC class 1 and MHC class 2 in detail. So what is the loci that produces MHC1? It's HLA-A, HLA-B, and HLA-C. All right. And did you notice this mnemonic? One letter. If you notice, all the HLAs which produce MHC1 or which codes for MHC1, they contain only one letter, HLA-A, HLA-B, and HLA-C. Now, if you look at the loci that codes for MHC2, they contain two letters, HLA-DP, HLA-DQ, and HLA-DR. Now, this is really high yield mnemonic, all right? The USMLE exam loves to ask you about these components. So you really need to know this. Now, let's look at the structure. Like I said earlier, MHC1 has a heavy chain and a beta 2 microglobulin, and this heavy chain is formed by alpha 1, 2, and alpha 3. All right? Now, compared to the structure of MHC1, MHC2 has two equal length chains, two alpha chains, alpha 1 and alpha 2, and two beta chains, beta 1, and beta 2. Let's look at their structure one more time. So this is the long chain or the heavy chain, which contains alpha 1, alpha 2, and alpha 3, and it contains a beta 2 microglobulin. Now, MHC class 2, it has two components, alpha and 2 beta. All right, alpha 1, alpha 2, beta 1, and beta 2. So this is MHC class 2, and this is MHC class 1. Now, let's talk about the binding that occurs between MHC and the T cells. Now, you need to know that MHC1 presents antigen to only CD8 positive T cells, whereas MHC2 presents its antigen to only CD4 T cells. All right. Now, this is super high yield. It looks like a simple information, but this is really tested on the USMLE exams. All right. MHC1 binds to CD8 positive T cells. MHC2 binds to CD4 T cells. All right. Now let's talk about expression. Which cells express MHC1 and which cells express MHC2? So you need to know that all the nucleated cells, antigen presenting cells, and platelets, they present MHC1 on their surface. All right, they all express MHC1 on their surface. But the unique thing is RBCs do not express MHC1. Now this is again a high yield information for exams. Now, unlike MHC1, only antigen presenting cells express MHC2, another high yield information. All right. Now, what is the function of MHC1? Like I've said earlier, its function is to present antigens to CD8 plus cytotoxic T cells. All right. Now, did you notice this mnemonic? 8 times 1 equals to 8. So, what does that mean? Now, you need to remember this mnemonic that the interaction between MHC and the T cells will give you a number 8. If MHC1 interacts with CD8 positive T cell, how you'll remember this is 8 times 1 equals 8. All right. The ultimate goal is to form number 8. That's our goal. We need to somehow get the number 8. Now, if you notice in MHC2, they present antigen to CD4 T helper cells. All right. Now, like I said, our goal is to form number 8. Now, what is CD4, right? 4 times MHC2. So what is 4 times 2? That's 8. So this is the mnemonic. MHC1 interacts with 8 to give you number 8. 8 times 1 equals 8. Now MHC2, 2 interacts with 4 to give you the number 8. All right? I'm saying you one more time. The goal is to form the number 8. If 8 interacts with 1, it gives you 8. If 4 interacts with 2, it gives you 8. All right. Now let's talk about antigen loading. Let's look at the figure. So let's talk about MHC1 pathway of antigen loading first. All right. So let's look at this figure. In MHC1, the first thing you need to notice is that MHC1 pathway presents intracellular antigen. All right. So when a cell is infected by a pathogen, it has different antigens in its surface, right? So these antigens are peptides, they're proteins, which are cleaved by proteasome into small peptides, right? The larger proteins are cleaved by the proteasome into small peptides. Now, these peptides are loaded into rough endoplasmic reticulum via TAP, which is transporter associated with antigen processing. All right. Once these peptides are loaded into rough endoplasmic reticulum, they're attached with the MHC2. Once they are attached to MHC2, they form a small endosome, which is then released into the cell surface, and MHC1 is able to bind to the cell surface of the. Now, after the peptides are loaded into rough endoplasmic reticulum, they bind to the MHC1, which is present inside RER, and then it forms a small vesicle. Now, this vesicle will bind to the surface, right, and release the MHC1 to the surface. Now, MHC1 will now finally be able to interact with T cell receptors of CD8 positive T cell and presents its antigen to CD8 plus T cell. All right, this is MHC1 pathway of antigen presentation. Now, let's look at MHC2 pathway. Now, this is slightly different. If you notice, in MHC1, it was intracellular pathogen, but in MHC2, the pathogen is extracellular. Now, this extracellular pathogen is first phagocytosed, all right? And then this phagocytosed pathogen will be destroyed into smaller pieces by a acid-filled lysosome. Now, this lysosome will come and fuse with the endosome and will destroy the pathogen, right? And then it will break different proteins into smaller peptides, which can be loaded. Now, once this acid-filled endosome interacts with the MHC2, the invariant chain will be destroyed, right? And finally, the antigen will be able to load into the MHC2 molecule. Now, after the loading of antigen to MHC2 molecule, this vesicle is finally able to fuse with the cell surface. Now, after the fusion with the cell surface, MHC2 receptors can present its antigen to CD4 positive T cell. All right. Now, talking about exam, there are numerous ways to ask a question, right? Sometimes there are numerous ways to ask questions from these two pathways. All right. Let's look at MHC1 pathway first. So the examiners will ask you questions like, there's a defect in TAP protein inside a cell. Which pathway of MHC presentation will be affected, or which T cell will not be able to recognize antigen, right? So we know that if there is a mutation in TAP, MHC1 won't be able to load antigen and present its antigen to CD8 plus T cell. So they can ask question in such pattern, or they might ask there is a problem in acidification of the lysosome, right? Which of the following MHC or which of the following cell will not be able to recognize antigen? If this acidification of the lysosome is affected, it cannot release the invariant chain. The only way to destroy or release the invariant chain, which is attached to MHC2, is by acidification. Now, if there's a problem in acidification, the invariant chain will keep on blocking the loading of the antigen into MHC2. So, the interaction between MHC2 and CD4 T cells will be affected. So, this is how they ask questions in USMLE step 1. All right. Now let's talk about T cell development and maturation. So T cells, they originate from lymphoid progenitor cells in bone marrow, but they mature in the thymus. That's why they are called T cells. All right, T for thymus. Now one thing you need to know that when they are formed in bone marrow, they do not have CD4 receptors or CD8 receptors. All right, these receptors are attached to T cells when they mature in the thymus. All right, let's look at this. Now, this maturation has two steps. They're called positive selection and negative selection. Now, let's talk about positive selection first. Like I just said, immature T cells which are formed in the bone marrow, they are CD4 negative and CD8 negative. All right, but they acquire CD4 and CD8 in the cortex of the thymus. All right. Now, thymic cortical cells also express MHC class 1 and MHC class 2, right? And if you remember from our video, MHC class 1 interacts with CD8 and MHC class 2 interacts with CD4, right? Now, what happens in positive selection of T cells in the cortex is that it makes sure that the T cell receptors can bind to the MHC appropriately. Now, this appropriately means neither too strongly nor too weakly, right? It has to be just the right amount. Now, the T cells, which has both CD4 and CD8, they're also called double positive, they receive a survival signal. That is, any T cells which contains both CD4 and CD8 can move to the next process of selection, all right, and maturation. Now, if there are some T cells which have some kind of abnormal binding with CD4 and CD8, they undergo apoptosis and they're destroyed. All right. We do not want T cells which have abnormal CD4 and CD8 receptors, right? Do we want that? Now, let's talk about negative selection of T cells in the thymus. Now, the positive selection occurred in the cortex, right? In cortex, they received CD4 and CD8. Now, in the negative selection of T cells, what happens is there is a test which occurs to determine whether the T cells bind to self-antigens presented on MHC by thymic medullary cells. All right. Positive selection occurred in the cortex, negative selection occurs in the medulla. All right. Now, like I said earlier, MHC class 1 presents its antigen to CD8 and MHC class 2 presents its antigen to CD4. Now, in negative selection, what we are doing is we are presenting self-antigens, antigens which are present in our own cells, right? We are presenting these antigens to the CD4 and CD8 positive T cells. Now, if there is a binding of T cells with self-antigen, that's pretty bad, right? If that binding occurs and if these cells are released into the bloodstream, they will cause inflammation and immunologic reaction against our own body. All right, and we don't want that to happen. So, if there is binding of T cells with self-antigens in the medulla, these cells are again destroyed. All right. T cells that do not bind receive a survival signal, but T cells that bind to self antigens undergo apoptosis. I know this is complex. That's why I want to explain everything once again by describing it in a figure. All right. So here comes the immature thymocytes. All right. They do not have CD4, they do not have CD8. They are double negative, right? CD4 negative and CD8 negative. Now, in the thymic cortex, they receive both CD4 and CD8. So they are CD4 positive and CD8 positive. Now, like everything, there might be some kind of mishap in this process, right? And we do not want abnormal T cells to mature. So any T cells which does not have CD4 and CD8 are destroyed, and T cells which have both CD4 and CD8, or also called as double positive, they are selected for survival, and they go to the next step in the thymic medulla. In the thymic medulla, negative selection occurs. So, in negative selection, it ensures that thymus does not produce self-reactive T cells. Now, in the cortex, they received both CD4 and CD8. Now we know CD8 interacts with MHC1 and CD4 interacts with MHC2 for antigen presentation. Now, there are numerous millions of antigens in our body as well. Do we want a T cell which can interact with our own antigens? No, right? We don't want that. It will cause autoimmune diseases. All right? Now, in thymic medulla, any T cells which binds to our own self antigens, they are destroyed. Now let's talk about antigen-specific T cell activation. Now, as we discussed, dendritic cells, they phagocytose antigens in different tissue, right? Let's say, for example, the skin. Now, after they phagocytose the antigen, the dendritic cell then moves to the lymph node, all right, the draining lymph node. Now, in the lymph nodes, they interact with T cell. Now, let's look at this example where it's interacting with CD4 T cell. All right. So how do I know this is a CD4 T cell? Look at this, MHC2, right? MHC2 interacts with CD4 cell. Now, this is a naive T cell. That means it has not been activated. All right. So this antigen on the MHC2 receptor will interact with TCR, the T cell receptors in the CD4 T cell, right? Now, this interaction between MHC2 and TCR is not enough to activate a naive T cell, all right? We need something called a co-stimulatory signal. And this interaction between B7 and CD28 is called co-stimulatory signal. Now, this co-stimulatory signal helps to prevent unwanted T cell activation. For example, if there is a self-antigen and if there is presentation of self-antigen to a T cell, this alone will not activate T cell. Our body needs another co-stimulatory signal to activate the T cell, and this is the interaction between B7 and CD28. Now, after this interaction between the primary signal and the secondary signal, there'll be release of cytokines from the antigen presenting cell. Now, depending on the type of cytokine, there'll be activation of different kind of CD4 T cell, which we'll be talking about later. Now, let's look at what happens if there is activation of T cell without a co-stimulatory signal. Now, there are things called super antigens, all right? These antigens are unique because they link MHC2 antigen presenting cell and T cell receptor and cause activation of T cells without the co-stimulatory signal. This is why they are called super antigens. And a common example is toxic shock syndrome toxin. All right. Now, this will lead to a severe inflammatory reaction, which can cause shock. Now, let's look at how cytokines released from antigen presenting cell will cause differentiation of CD4 T cells. Now, I have included Th1, Th2, Th17, and T regulatory cell in this image, but you need to remember that Treg cell is not a type of T helper cell, all right? It is not a CD4 T cell, but there is a huge importance of T regulatory cell. That's why I have included this in the image. But there are mainly Th1, Th2, Th17, and follicular T cells, which are the main subtypes of CD4 cell. All right. Now, depending on the cytokines, if the cytokine released from antigen presenting cell is interferon gamma or interleukin 12, it will cause differentiation of CD4 cell into a Th1 subtype. All right. And this Th1 subtype will produce interferon gamma and TNF alpha. Now, this is really important. Now, from exam point of view, how they will ask a question is, in infection, macrophage and cytotoxic T cells were activated. Which of the following cytokines were released which induced the activation of macrophages and cytotoxic T cell? And in that situation, the right answer could be either interferon gamma or TNF alpha. So this is how the question will be asked. Now, this is a high yield table, all right? There's one more type of question they ask. There was activation of Th2 cell. Which of the following interleukins activated Th2 cell? Now, I'm oversimplifying the question, but you get what I'm trying to say, right? Now, you need to know which cytokines activate these T helper subsets and what are the cytokines produced by the subsets itself and what is their function. All right, this is super high yield table and you need to memorize each and every interleukins. But we'll be talking about interleukins and their function in a separate video, but this is a gross overview. All right. So once Th1 cells are activated, they will release interferon gamma, TNF alpha, which will activate macrophages and cytotoxic T cells. And this is really important for granuloma formation. All right. Macrophages play a huge role in producing granulomas. Let's talk about how Th2 cells are activated. If there is release of interleukin 2 and interleukin 4 from the antigen presenting cell, the CD4 cell will divide into Th2 subtype. All right. And what is the function of Th2 cell? It will secrete interleukin 4, interleukin 5, interleukin 9, and interleukin 13. Now, the role of these interleukin are to activate eosinophils and produce IgE. All right. So they help in IgE class switching. We'll be talking about class switching and immunoglobulins in a separate video, but you need to know what activates IgE formation, and it's interleukin 4, 5, 9, and 13. Now, like I said earlier, you also need to know what activated Th2 subtype. It's interleukin 2 and 4. Now, the mnemonic is 2 times 2 is 4, right? 2 times 2 is 4. So, interleukin 2 and 4 will activate Th2. Now, the mnemonic for Th1 is, if you notice, this interleukin 12 has a 1, right? So it will activate Th1. Now, let's look at what happens if interleukin 6, interleukin 23, and TGF beta are secreted from the antigen presenting cell. These interleukins will activate Th17 subtype of the CD4 cell. All right. So what is the function of Th17? It will secrete interleukin 17 and 22, which will induce neutrophilic infiltration. So their role is to induce neutrophil infiltration. Now the mnemonic for this is, if you subtract 6 from 23, you'll get 17, right? So interleukin 6 and 23 will activate Th17 subtype. All right. Now, the function of Treg is to maintain tolerance. or maintain immune tolerance, all right? They prevent excessive activation of T cells. They downregulate the inflammation by suppressing the activation of T cell. And once an infection is controlled, once the body recognizes that the inflammation needs to be reduced, there'll be production of interleukin 10 and TGF beta, right? Now, this interleukin 10 and TGF beta will cause the Treg cell to be activated. And once Treg cell is activated, it will again produce interleukin 10 and TGF beta, which will cause positive feedback inhibition. Now, Treg cell will itself produce interleukin 10 and TGF beta, which will cause feedback loop, right? Positive feedback loop, and help to maintain immune tolerance. This is really important for preventing autoimmunity, all right? If there is unwanted activation of T cell, body will try to produce interleukin 10 and TGF beta, which will try to downregulate the autoimmune response. Now, as I mentioned, cytotoxic T cells in this figure, let's talk about cytotoxic T cells. Now, the function of cytotoxic T cell after activation by Th1 cell is to cause apoptosis of virus-infected cell, neoplastic cell, and donor graft cells. They release cytotoxic granules containing perforin and granzymes, and they destroy these infected and abnormal cells. Now, like I said earlier, cytotoxic T cells are further activated by Th1 cells. All right. Now, as we talked briefly about regulatory T cell in the earlier slide, now let's talk about regulatory T cell in further detail. Like I said earlier, Treg cell maintains immune tolerance by suppressing T cells, right? They suppress CD4 and CD8 T cells. Now, how do you recognize regulatory T cells? They are recognized by expression of CD3, CD4, CD25, and FOXP3. So these FOXP3 and CD25 are unique to regulatory T cells. Now, activated regulatory T cells produce anti-inflammatory cytokines like interleukin 10 and TGF beta. So what happens if there's dysfunction of Treg cells? Mutation in FOXP3 leads to unregulated T cell activity, and it is associated with IPEX syndrome. The full form is immune dysregulation, polyendocrinopathy, enteropathy, and it's X-linked disease. All right. So, what are the clinical features of IPEX syndrome? It's endocrinopathy. That means there could be diabetes mellitus, hypothyroidism, and other endocrine diseases, right? The hallmark feature is enteropathy. In immune dysregulation caused by dysfunction of Treg cells, there'll be chronic diarrhea because of autoimmune enteritis. Now, besides endocrinopathy and enteropathy, you might see nail dystrophy and eczematous dermatitis. Now, if you perform a biopsy of the intestine, you might see lymphocyte infiltrate and atrophy of the intestine, right? Now, like I said earlier, as there'll be endocrinopathy, the presenting feature could be diabetes in infants. Now, as it is an X-linked disease, only males are affected, all right? Now, let's look at the activation of CD8 plus T cell. We have talked about CD8 plus T cell, which is also called cytotoxic T cell, numerous times in this video, but let's look at this image one more time to consolidate our concept. So the activation of CD plus T cell is exactly similar to CD4 T cell, but there's only one difference. Instead of MHC2 presenting the antigen to CD4, in CD8 T cells, the antigen will be presented by MHC1. All right, that is the only difference. The dendritic cells will present the antigen via MHC1. Let's talk about B cell formation. Unlike T cell, which was formed in the bone marrow and matured in the thymus, B cell formation and maturation occurs in the bone marrow itself. So once a mature B cell is formed, it will be called naive B cell. It's mature but naive. That means it has not been exposed to an antigen. Now, the unique thing about B cell is that it expresses CD19, CD20, CD21, and CD40 receptors. And they also express MHC2 and B7. All right. So the unique thing is CD19, 20, and 21. This is super high yield. in exams, sometimes they will not mention that the cell is B cell, but they will mention something like the cell is CD19 positive, and you have to understand that they are talking about B cell. All right. Now let's talk about B cell activation. Now, the unique thing about B cell is that it has an ability to endocytose an antigen and present that antigen via MHC molecule, MHC2 molecule to be precise, and activate follicular T cells. All right. Now, once antigen is presented by MHC2 to the follicular T cell will be activated. Now, it will release cytokines which will activate the B cell. All right. Let me repeat it one more time. B cell will endocytose an antigen and present that antigen on MHC2 molecule to the T cell receptors. These T cell receptors are in T follicular helper cells. All right. It's a type of CD4 positive T cell. Now, this presentation will activate follicular T cells. Now, once follicular helper T cells are activated, it will produce interleukin 4, 6, and interleukin 21. And these cytokines will activate B cell. Now, the interaction between MHC2 molecule and T cell receptors is not enough for activation of B cell. There must be interaction between CD40 ligand and CD40 receptor. After the activation of B cell by different interleukins like interleukin 4, 6, 21, and the interaction between different receptors like CD40 ligand and CD40, and the interaction between MHC2 and T cell receptor, B cell will be activated fully, right? And it will undergo proliferation. Now, B cells are activated and they produce IgM. This happens initially. The initial immunoglobulin which is produced by activated B cell is IgM. Now, depending on the cytokine produced, there'll be different type of class switching and production of different type of immunoglobulins like IgA, IgE, and IgG. And there'll be affinity maturation. Now, these topics can't be covered in this video, and we have a separate video for class switching and affinity maturation. Now, after the activation of B cell, there'll be production of two types of B cell, plasma cell and memory B cell. The function of plasma cell is to produce immunoglobulins, whereas the function of memory B cell is to recognize the specific antigen which triggered this particular B cell to recognize the antigen in the future as well. Now, if this antigen is exposed once again in our body, in the future, there'll be robust activation of B cell and there'll be quicker production of immunoglobulins. Now, time to do some USMLE style questions. So let's look at this question. A 1-year-old boy presented with a history of chronic diarrhea, failure to thrive, and recurrent pneumonia. Genetic testing reveals a mutation of a receptor on a lymphocytic cell. In order to activate this lymphocyte, APC presents antigen via MHC2. Further testing revealed that this lymphocyte is CD19 positive. Which of the following receptor is most likely mutated? Now, like I said earlier, this cell is CD19 positive. Now, if you remember what I told you, CD19 positive means this is a B cell. All right. Now, we talked about activation of B cell, right? Now, let's look at the option. CD40L means that it's a ligand, it's not a receptor. So we can rule that out because the question is asking which receptor is mutated. So it's definitely not A. Now, our options are CD40, CXCR4, and CD80, 86, right? Now, if you remember what I told you, the activation of B cell is dependent on interaction between CD40 ligand and CD40 receptor, right? So, in this question, there's mutation of CD40 receptor, which caused failure of activation of B cell. All right. Let's look at another question. Some scientists in a lab decided to prevent acidification of lysosomes within antigen-presenting cells in an experimental setting. The modified cells show impaired interaction with T lymphocytes when exposed to an antigen. The observed effect most likely results from a low surface expression of which of the following molecule? Is it MHC class 1 or MHC class 2, T cell receptors, or toll-like receptor? Now, if you read the question carefully, it says there is problem in acidification of the lysosome, right? The scientists, they prevented the acidification of lysosome. Now, if you remember from the video earlier, the acidification of lysosome is really important for MHC class 2. All right, so the answer is MHC class 2. Now, let's look at the third question. A 2-year-old boy was diagnosed with type 1 diabetes mellitus 2 weeks back. He has also had chronic diarrhea, failure to thrive, dermatitis since infancy. Genetic testing reveals a missense mutation affecting FOXP3. Which of the following is most likely cause of this patient's condition? Now, we talked about this earlier, right? The mutation in FOXP3 will cause IPIC syndrome, and the patient has classic feature. The child is a boy, that is X-linked, right? The patient has type 1 diabetes mellitus, there's endocrinopathy, there's chronic diarrhea, which stands for the E, right? The E in the IPIC stands for enteropathy, and this patient has chronic diarrhea. And he also has dermatitis, which is a feature of IPEX. So this is a dysfunction of regulatory T cells. All right. Now let's look at the fourth question. A 9-year-old female child is brought to her pediatrician's office by her mother, who appears really anxious. The child has had a runny nose, sore throat, cough, low-grade fever of 100.2 degree Fahrenheit for the past 2 days. The doctor explained that these are features of a viral illness and reassured the mother. The doctor explained to a medical student who was listening that the cytotoxic CD8 plus lymphocytes are able to kill virus-infected nasal epithelial cells, right? And these cytotoxic CD8 plus lymphocyte receptors recognize foreign proteins presented on epithelial cell surface by MHC molecule. These MHC molecules comprise of which of the following components? Now, this sounds like a tricky question, but it's pretty easy. Now, if you remember my mnemonic, MHC class 2 interacts with CD4, right? 4 times 2 is 8. Here in our question, the CD8 plus T cell are interacting with the MHC molecule. Now, 8 times 1 is 8. Our ultimate goal is to get the answer 8, right? In this question, the CD8 positive T cell will interact with MHC1, right? So this has to be MHC1. Now, is that knowledge enough to get this answer correct? No, because all the options have MHC class 1, right? So now you need to know the structure of MHC class 1. So let's look at the option MHC class 1 heavy chain and IgG, right? MHC class 1 heavy chain and beta 2 microglobulin. MHC class 1 heavy chain only, and MHC class 1 heavy chain and IgN. Now, if you remember what I've taught you so far, the components of MHC class 1 includes beta 2 microglobulin. All right. So the right answer is B, MHC class 1 heavy chain and beta 2 microglobulin. All right. So the right answer is MHC class 1 heavy chain and beta 2 microglobulin. Thank you.